Ich habe die wunderbar... Ich habe die Ehre, den Abend heute zu eröffnen. Ich versuche mich so weit es geht kurz zu halten. Ich bin Rüdiger Wassibauer, Schmiede allein und Inselzeit. Ich freue mich heute sehr, ein neues Format begrüßen zu dürfen, das Claudia mich herangetragen hat. Und was ja auf eine Stammzellen-Research und Artist in Residence-Programm zurückgeht. Anna wird sicher noch länger darüber sprechen und auch Claudia. Und ich hoffe, es geht hier in erster Linie um Kunst und Wissenschaft. Und wir bedanken uns dafür, dass wir hier sein dürfen. Wir bedanken uns besonders beim Land Salzburg, beim Bundesministerium und bei der Stadt Hallein. Und in diesem Moment würde ich ganz kurz das Mikrofon an die Frau Rosa Bock weitergeben. Das Mikrofon ist an, aber es geht nur auf das Video.
Also, da muss ich es also doch herhalten. Ja, ich darf Sie als Kulturstadträtin von der Stadt Hallein recht herzlich begrüßen. Rüdiger Wassibauer hat mich schon vorgestellt. Ich werde zum Inhaltlichen gar nichts dazu sagen, aber ich freue mich, dass sowohl die Schmiede als auch die Inselzeit schon sehr lange bei uns in Hallein zu Gast ist, vor allem die Schmiede mehr als 20 Jahre, die Inselzeit bereits das 4. Jahr, und wir sind stolz, dass wir so internationale und hochkarätige Künstler mit kreativsten Ideen bei uns begrüßen dürfen. Ich mache es kurz. Ich wünsche einen guten Abend und ein gutes Gelingen für das gesamte Festival. Dankeschön.
Die Übergangsbrücke, danke, aber international. Und jetzt werden wir in Englisch weiterfahren. Liebe Claudia, bitte gib ein bisschen auf die Ausstellung.
Thank you, Rüdiger. So I'll immediately start in English. Thank you for Kulturstadträtin for joining us and for inviting and hosting us here. Thank you, Rüdiger, for hosting us at Inselzeit. And thank you to Helmholtz Munich for supporting also this project. My name is Claudia Schnuck and I'm a curator. I'm working at the intersection of art and science. So what I normally do is I bring artists to work with scientists, and this was actually also what we did in this project, I think in 2019. Maria Elena Torres Padilla, she's the director of the Institute of Epigenetics and Stem Cells at the Helmholtz Zentrum Munich, approached me and she said, oh, she's so excited by all these art science projects. We have to do something and we have to host an artist in her lab. And you have to imagine her lab is quite big. She hosts 4 groups and has quite a few scientists working with her on different aspects on epigenetics and stem cell research. And as scientists, they're working with, starting already with these words on very abstract topics for most people, but they also use quite a few very interesting and poetic words like cell memories and cell fates, and how also this is related to the origin of life. So we made a small call for artists, and we invited artists to propose projects. And this is how Anna Dumitriu, our artist from Great Britain, came into the play. So we started in beginning of 2020 to work with her, where she proposed already the title inspired by the cell memories and the cell fates, the mutability of memories and fates, to investigate and work with a different research group at the Helmholtz Zentrum Munich. And I think from there, it's been now a 4 years or even longer journey, because you all know what happened in 2020. So we started the discussion beginning of 2020. We had the first research or artist in residence visit planned for May 2020, which could not happen. So we started slowly online. And Anna had later, was it still 2020 or beginning of 2021? The first actual visit in the lab. And from there, actually, all the works that you see in this exhibition developed. And we are happy to, for the first time, show all the outcomes of this residency, of this fruitful interaction between the scientists and Anna, because we also have, so the residency went on and on in a collaboration. And the last few pieces just were finished this summer. So you see also quite a few of premieres in this exhibition, because I think 6 of the works have not been exhibited yet. So from there, I think I also will quickly introduce the artist and the scientist, and here we then also start into the discussion, what's the background of the project. So now I start with Anna. Anna Dumitriu is a bioartist, so she's internationally well known for an award winning for all these intersections, specifically working with microbiologists. I think she's been doing this now for 25 years nearly. So she's really an expert in working with scientists, but also working in labs. So without the biology education, she really started to become also proficient in lab work and talking to the scientists, but also drawing from the different information that she gets, or the knowledge that she accumulated over the years in the different labs to draw the lines between the research. And Maria Elena Torres Padilla, as I said before, she's at the Helmholtz-Zentrum Munich, director of Institute of Epigenetics and Stem Cells, but she's also professor at the university in Munich in this field. And I think so many other things that you're the director of that I cannot repeat all of them. But yeah, I will stop talking now, and maybe I start with you, Maria Elena, why you came up with this inspiration that you really wanted to work with an artist, and how this dialogue with Anna also unfolded.
I thought I also should try my 2 pairs of words in German. Herzlich willkommen. Von meiner Seite auch. Yeah, and I'm actually very excited to be here and be part of this exhibition. This is the first time that we are showing all the pieces that Anna has done as a result of the work, as Claudia said. And I guess that I was really very attracted to do this work together with an artist that ended up, luckily for me, being Anna. I think because I'm really fascinated by the research that I have been doing for the last almost 20 years also. And I thought that it could be a great opportunity to learn from the creativity of a colleague, artistic colleague, on how they could embrace this scientific question, but also to share the knowledge, the passion that really, you know, we go to the lab and we do these crazy things. And I thought that this could be a great opportunity for me to learn from a completely different process of creativity and expose the science that we do through the eyes of an artist. And obviously I'm very happy that this has led to a number of very exciting works that I hope that you will also enjoy this evening.
You want to go on?
Yeah, I mean, so yeah, 2020 was an interesting process because we had to pivot to doing it completely online. And so what we did was I had meetings with scientists from all the labs, lots of different scientists, for like a whole year. So I was, we were having like maybe 3 or 4 meetings a week, like 1-hour things where people would explain their research to me. So it was quite a steep learning curve because, as Claudia mentioned, I've worked a lot with bacteria. I work a lot in labs. I'm very into microbiology, but I hadn't worked so much with stem cells. But I've always had an interest, and I wrote my master's dissertation on immortality, and I've done work about the sort of science behind ideas around immortality and things like that. And kind of one of the things with stem cell research is to ultimately to make spare parts, like to understand how cells work and then build somebody a new heart. I mean, we're a long way off, but because the heart's made up of lots of different cell types and things, but theoretically in the future it would be possible, we hope, maybe, or not, to maybe 3D print a human heart that could actually be made from your own cells and create spare parts. So there's a tie-in to this idea of immortality there, and that was kind of deep in what I was thinking about. And then it was this idea of the mutability of memories and fate. So the idea that whether cells have a memory of the cell that they were before, because they can sort of wipe the memory of a cell, and that's how you make it's called an induced pluripotent stem cell. So you'll see it written on some of the slides out there, iPSC. And that's, so these stem cells, they can make almost any cell type in the human body, except for placenta, which a totipotent cell can make. But these stem cells can make anything else. But they have to, I mean, and then you get into the complicated thing of they apply certain factors to them to direct their growth into whatever cell type. And so when you can get stem cells and that are kind of blank, and you can apply these factors to them, and you can turn them into heart cells or something like that. So that was fascinating to me in I guess at the start of the process. And this idea of the fate is what it's going to be. Like, how does a cell know so there's a picture out there of the DNA structure. It's not the DNA structure, the chromatin structure of the DNA. So DNA is a double helix, we know that, but it's got all this stuff attached to it, and it's wrapped around things, and there's all sorts of stuff, whether it can be modified or not. And in the picture out there, we've got an image that we took in the lab with Adam, who's here, of the moment. It's a zygote, so it's before the mother and the father cells have merged even in an egg. So we've got that stage. And then how do they know when they start dividing? How do they know where to go? How do they know what to be? I'm an arm, I need to go over there. I'm going to be an eye, I need to go up there. So that's kind of the stuff that you're kind of studying. How does that work? So it's all really fascinating stuff that I just basically took a deep dive into, and hopefully I can share the sort of ideas behind it with you with the artworks.
You want to add something a bit about the scientific background and also how you came to work with cell fates and cell memory, and also how you connect to Anna's questions?
Yeah, sure. I think that for me what has been fascinating is that if you think about your own body, you have all these different cell types, right? Your skin cells will protect you from injuries, your heart will beat and make you having the blood. But they all come from one single cell, which is basically the result of the egg and the sperm at fertilization. And so if you think about that, from one single cell, you give rise to all the millions of different cells that make your body and that allow us to function, to breathe, to eat, to do all our daily stuff. And that cell has the same genetic material. So the DNA is exactly the same throughout all your cells, with some exceptions. And so for me, the question is, how on earth can we make all these different cells that make our body so complete and so complex from one single genetic information? And so that has been the subject of research. I've been fascinated. And it's very linked to this process of memory, right? How does the cell that I divided, it's almost imagine like if I am a cell and I'm coming from a cell that has a different fate or a different identity. If I want to become now a cell in the skin, how do I rewind my memory and say, no, now I have to use these genes because I need to change. So that's very linked to this. I mean, I learned after working with Anna that this is a very romantic concept of fate, right? Where will you end? And for me, understanding the molecular basis of that, because this is really how life starts, right? We all were one cell at that point. So that's what drives me, and that's the scientific vision that we have been working on really almost 20 years. And of course, there's a lot of molecular complications. And I talk a lot, and I can talk for 4 hours, but I don't want to bore you with the molecular complications. I just think it's a beautiful process that we sometimes forget, right? That is really our everyday life. And so that's basically where we have been devoting our research on that. And I think it's beautiful to see that it has a lot of implications of how we perceive our functions and how we are interacting with the environment as well.
But Anna, so coming from that, so this is actually, as Anna said, also a very romantic conception of your research topic. This kind of poetic questions also have other implications and other layers of meaning. And this is actually where you also always are very strong in bringing together these different ideas. So you're not just taking, of course, you're taking some of these beautiful imagery, you're taking some of the very specific scientific information that the scientists provide to you, but you also engage yourself in projects directly in the lab. We did some experiments that you proposed, and you also tried then to go even further and connect to cultural history and to everyday life aspects, so that in the artwork you have all these different layers of really scientific information, but also everyday stories. You want to talk a little bit about your process?
I think what I try to do is I try to get as much as I can from the scientists who are explaining it to me. And I think one thing I pride myself with is asking stupid questions, because if I don't understand it, I have to ask and I ask and I ask, and I don't leave it, do I? I don't leave it. It might get a bit annoying after a while, but I want to understand it properly. And one of the things of doing the experiments hands-on in the lab is me understanding through the physical process of doing it, which I think is a much better way of learning and grasping stuff. So that's why I like to make the artworks hands-on as much as possible in the lab. But then I think a lot about metaphor as well. So we were talking about this chromatin structure. So there's the necklace out there, which you can see with the copper sulfate, and that's representing the chromatin structure. So this is the way that the DNA, which is just the little thread that's wrapped around these blobs. The blobs are called histones, and the DNA is wrapped around them. And I decided that we would make the chromatin structure of the OCT4 gene, which is one of these Yanamaka factors that's always present in stem cells. I keep looking because in case I get any of the science wrong. And then these ribbons that are tied together, that tie everything together, these are representing the H3K9 trimethylation. It's right, isn't it, Adam? Yes, good. Took me ages to learn that. And so this is like how the structure is knotted together, and when it's knotted together like that, it can't be modified by epigenetic factors. And when it's loose, like the orange histones, they can be modified, so they can be changed. So we went somewhere in between the two, and then the whole piece is impregnated with the OCT4 gene that we extracted in the lab. And then the reason I use copper sulfate is in the first experiment I did in the lab, we added copper sulfate to the experiment. I'm always looking for the materiality of things. What can I use in the work? And why do we use copper sulfate here? Oh, it's important for the cells to grow. They need a bit of it. So that's why there's this copper sulfate, this blue theme through the work as well. And then there's the knitting.
Maybe we stay a bit with the necklace, because for the necklace there was also this, was it also Adam explaining it to you, like the beads on a necklace with certain distance? And I think you had quite a few really interesting conversations also about the necklace with all the scientists, or quite a few of the scientists, getting the details right, but also then working it somehow in a small workshop together with the scientists to really understand what is the correct translation into this tangible object. You want to follow up on that?
Yeah, I think I mean there's a lot of beautiful details in nature, obviously, because as I said, but they are reproduced and taken into Anna's work. And the case of the necklace, which is the piece that you will see in the middle, it took actually quite a bit of back and forth. As Anna was saying, she just asking, but you know how it is. And then you think about the DNA, and then this is thread, but then it has to be wrapped around specific proteins. And then Anna said, I want to do that gene. And then I said, yeah, just put the DNA around this thing. Now, how many proteins you have? what's the size of the DNA? So that process was actually quite exciting, because also as a scientist, we realized that there's still a lot of things that we don't know. And she was asking questions about, how many proteins do you need to have all the DNA so that that gene can function? And a lot of this we didn't know, or we had to tell it, we have to go and look in the publications, or we have to do the experiments. And so I think that work of trying to tell you all today also the details about how come, it's quite exciting for me, because this is also a little bit of the way we approach our science, and we want to understand how this gene, which is essential for stem cells, if you don't have that gene, you can't have stem cells, and therefore you can't have your bodies. And there's a lot of nature wrapping around those that has been retaken all by Anna's work. So.
I would add as well that in nature the DNA wraps around the histones magnetically. It's like, so it just loops and it goes there. And when you want to make it physically, you actually have to wrap it round kind of by hand. But that's all very well. But the way it wraps is you have to wrap one and then draw it to the entire full length of the strand. So you need to start with the length that you want ultimately, and then you need to wrap each one along each. It's really fiddly, and that in essence is why we did it as part of a workshop, because I made one and it was very hard work at home, and I thought, well, we could make some more, we could do a workshop, so that'd be fun for everyone, and I share the work. So that's what's nice about working with a big group of scientists as well, because we can all work together as well. And Adam helped with that workshop and brought all the science to it as well, so that was lovely.
I interrupted your description of the knitting piece. Do you want to go on with the knitting piece, or you want to go on to a different element, also maybe about experiments?
Well, I could say a bit about the knitting, because it's quite a good thing about explanation of cell memory. Because when we were talking about, so if you've got this tightly wrapped around chromatin structure, you want to, as you make a stem cell, you've got to unpick it basically. And I was like, oh, so it's like a jumper. It's like when if you ever done a thing where you wanted to make a new jumper out of an old jumper, and that new jumper kind of has the memory of the old jumper. It's all crinkly and it takes ages to get those kinks out. And that's kind of how it works with the wiping the memory of the cells, making them stem cells. Is do they still have those kinks like the jumper? So I decided to make this piece with the squares and then shrink it a lot and then unpick it and remake it. I'm not very good at knitting, so there's two versions of this work. One's made by my daughter-in-law, and this beautiful one here, which is the new and improved version, is Claudia did all the knitting for it. I ultimately I like to collaborate with people, share the workload of things I'm not very good at, and then I darned it, because the DNA obviously sometimes when it needs repairing. It has problems with the way the DNA is transcribed. So the visible mending that I did on there is a representation of that as well, and then that is also impregnated with the DNA of the OCT4 gene again. Yeah.
And maybe we go to a little bigger body parts, and this wiping the memory and going back to stem cells and making different cells. Maybe you also want to talk a bit about the main piece, so the piece called The Mutability of Memories and Fates, which is actually a premiere today to see it all. Also the full trilogy of works that coming together. So there's this question, do cells have memory? Do they remember the cells they were before? And I said, well, maybe we could try and see if they did. Why don't we get some stem cells and make them into one cell type, and then take them back to stem cells and then turn them into something else? And for some reason I wanted to do it 4 times. I think it's to do with the seasons or something. I was thinking of the seasons of life. But what if we could do that 4 times, and then we'd know? Because if they have memory, then they if they do remember being a heart cell and they've gone back to a stem cell, if they have the memory, then how are they going to want to be those if you used exactly the same cells?
and turn them into something else? How would that work? It just wouldn't work, would it? So I proposed this experiment, which I remember the moment, because Adam said, this is very ambitious, which he's a master of understatement. and so, yeah, that's what we decided to attempt to do. And
you were also very excited about this.
Yeah, I mean, this was quite surprising, because I realized that no scientist had actually thought about that before. So we had to wait until Anna came and asked a simple question, right? Because I told you before, your cells have the same DNA. We can change the fate. Theoretically, we can go back and forth, right? As many times as possible. But this has actually not been addressed. And so there was a significant amount of time spent on trying to see, can we actually do it, right? Is what I'm telling you correct? You have the same DNA, you go to the lung, for example, and then you revert back to this stem cell. And this is actually something that is done in hospitals when you give cells for therapy. But then can we then get that stem cell to make a heart or a heart cell and then back? And how many times can we, right? Is this really as flexible? So I remember having this conversation with Anna, and then I was sort of speechless. I said, I don't know. No one has actually done that, and I agree. This was ambitious. We did some. I mean, I didn't do the work. Some people in the lab did the work with Anna, and the result of some of that work you'll see over there later on. So very exciting thinking about those possibilities from the experimental side of things.
So yeah, I mean, and one thing I was told was, like, there's one lab that does the stem cell to heart cell, and they've published that, and then there's another lab that's done heart cell to stem cell, and they've published that. And so the piece is kind of bringing together all that different research from all those different labs, and seeing and trying these protocols that you gathered to see if it would actually work. And sometimes it didn't work, and then, like, why didn't it work? And then going back to square one, square two, wherever we were. So the cells have been stem cells, then they became neuronal progenitor cells, which is basically brain cells, back to stem cells, then to heart cells that actually beat in culture, so you can see them actually beating. What we noticed about those is they beat irregularly. This happened when we were doing the soundtrack for the piece. We noticed the cells aren't beating exactly in time, so we had to edit the heartbeats that you hear to go slightly off time. We don't know why, so it's another question that we came up with today. And then they go to back to stem cells, then to hepatocytes, which are liver cells, and then back to stem cells, then to lung cells, and finally back to stem cells. And I'd like at some point to turn them into totipotent cells that can become anything. But the research hasn't caught up with my artistic desires yet.
We need to do some more work.
Yeah. So just to kind of put the artistic perspective on this, I've always been, like, fascinated in notions of immortality, and this idea of the spare parts. And if we were immortal, I get a little bit geeky here, because I'm quite interested in sort of vampire mythology of, like, and literature around that. So I wrote my master's dissertation on sort of the science of immortality, but also how it's represented in popular culture. This is a long time ago. And some of these, like, representations of vampires as, like, they've lived thousands of years or hundreds of years. What do they do with this time? I'm quite inspired by Anne Rice's books. I don't know if you've come across this. There's a vampire in it who lives like 300 years and he's never seen the sunrise. He lives long enough that he can go to the cinema and see the sunrise and watch that. Or there's some other vampires in the same series where he's lived for like 500 years or 1,000 years, and he realised that the rock stars look like him, so he becomes a rock star. And if we were, and there's the original vampires in the story, they just sleep. They don't want to do anything much anymore. They can't think. They've run out of ideas, really. They just mostly sleep. And what would, if we could live these immortal lives, what would we do with those lives? Would we be artists for a bit? Then maybe be a scientist. Maybe I want to be a curator. Maybe I always think maybe I want to be a lawyer at some point, and then you'd be like, oh, that was a bad idea, or something like, or be a doctor. Then maybe you'd sleep. I don't. It's a really interesting question of like, if we were immortal, what would we do with that time? So it was like these cells have lived 4 different lives, and I was thinking like, what if we could live these different lives? So that's the idea behind it. And accompanying the video of the actual cells and their transformations, there's a kind of genie bottle out there, and that's got a vial of the cells in it. They're dead now because they're not in the freezer, but there is another vial of cells in the freezer at the Helmholtz Zentrum that has lived 4 different lives. And there's some microscope slides of each flip that they've done is out there as well, so you can see those.
And then there is one more component. We've been talking a lot about stem cells, but what about the epigenetics? So there is one more piece also that is also more recent work that's still a work in progress, but it's also based in this idea of epigenetics. And maybe you want to start a bit with the epigenetics part.
Yeah, I could say that epigenetics, I mean, I can again speak for 4 hours, but I don't want to bore everybody. I just probably will use the same explanation I did before to tell you that all our cells are the same genetically speaking, but they do different things. And that's because of epigenetics. So it's sort of epigenetics above genetics, so that's Greek. And so that's based on the genes, but depending on how you modify this genetic material, cells can do different things. So we are the result of pure epigenetics. And that's what allows us to actually change the fate of the cells without changing the genes. You just change the above the genes, but not the genes. And so that's also why I'm fascinated by epigenetics, because it's also reversible, right? And I think a lot of people at some point 20 years ago were talking, once we figure out the genetics and the complete genome, and there's all this money spent on sequencing and so on, we will understand life. Well, it turns out that we underestimated that epigenetics is also important for that. And so that's really the conceptual basic of the work that we do to understand how you can change the cell fate, even though the DNA is the same.
And epigenetics is also known a little bit to a broader public, but not only in this very concrete explanation as you just put it, but also about how external influences also then might influence the next generation. And this is something that you are working on right now in the next project.
Yeah, so we talked about it a lot through the project, but I didn't get there we're doing at the time, so it's a next step thing. And I was like, so what if we change what things are epigenetic effects? What, like, if I'm in a certain environment, like, how would I look different? You know, like what would be affecting me, you know? And I thought about making like a 3D printed model of something of me with, you know, different epigenetic effects, but I didn't really like the idea. What I like to do, ideas just sort of rattle around in my head until the moment that they're really fully formed, and I knew that wasn't fully formed, and it is still not fully formed, this idea. But what I was so I met some researchers at King's College in London, and they have a very big department of war studies there, and they were offering seed funding for projects, and I told them about the research that I learned about in the lab, which was from the Dutch hunger winter. Have you heard about this? It's so from 1944, just I'll explain it briefly for those who haven't. From late 1944 to early 1945, the Nazis blockaded the food supplies in the Netherlands to punish them for a rail strike, which was basically to help the Allies to invade again. So so they were punished, and at least 18,000 people died, maybe more. But what was unusual about it is that they were all well fed and healthy before this moment, and then as soon as the famine ended, they were again well fed and healthy. So it was a very sort of discrete, clear point. And then researchers found that babies that were in the womb up to 12 weeks as a fetus, when they were born, which was at perfectly normal weight and there was nothing strange about it, they went on in life to develop type 2 diabetes more. They have the genes for type 2 diabetes and also slower metabolism. So their metabolism is sort of downplayed, downgraded. And this is because of an epigenetic effect, that they were basically born into a world of famine, and then there wasn't a famine. Because so this is like the first and main sort of scientific case of this. And what was fascinating as well was you told me that also the female babies that were in the womb have the eggs for the next generation as well inside them. So this epigenetic effect can be at least passed down to 2 generations. So I wanted to do work about that, and I was talking to Professor Rachel Kerr and another curator called Cécile Bonfarrier, who are in the War Studies department, and they brought this idea of post-memory to it as well, this way that memories are kind of passed down and altered. So it was, again, this mutability of memory and how memory can be passed down through generations, like stories my mother told me of World War II or something. I never experienced it, but I sort of think I know a lot about it, but it's not my memory. So it's this sort of thing. So what you can see out there at the moment, and this is a very early prototype, it's a spoon, basically. It's a 3D printed spoon, and it represents the spoons that the children would carry during the Dutch hunger winter. Apparently they all carried a spoon with them in case they found a soup kitchen that actually had something to eat. So they would all carry their spoons. Audrey Hepburn was one of these, the famous film star, was one of these children at the time, because her mother decided to move to the Netherlands because she thought it would be safer during World War II, and so she experienced this. And famously, because they couldn't ship out tulip bulbs for the same reason they couldn't get food in, they ate tulip bulbs. So this was something, tulip bulb soup and all sorts of things made of tulip bulbs. And they ate Swedish flour, which was then one of the first things to break the famine, that came on Swedish boats, enough for half a loaf of bread for each citizen. And they ate chicory root, they ate sugar beet root, the actual roots. They ate cats, they ate dogs, all sorts. They had nothing. So the spoon is a tulip petal, and at the end you can see the tulip bulb, but inside there is the 8-week fetus that might be being affected by it. And so where we are at the moment with this project is that we're trying to do the next stage of funding for it and move on. I've done loads of research on it, and then I want to make some bigger artworks, and we want to do like food related projects or like workshops, and we want to plant tulip bulbs, and I want to make some more 3D printed objects, like a kind of monstrous tulip that has these effects to it. So all 3D print. I like to print the spoon in metal, which I can't afford to do at the moment. So and the beans round it are fava beans. They're Dutch field beans, which would be the brown beans that they ate in the soup in those days. And I've done another project on that, weirdly, so I knew about that. And they were believed in kind of in mythology, I guess, back in ancient Greece. They were thought to be like symbols of embryos. So they look like embryos, so that's why the beans are there.
And we're going to have a workshop tomorrow with you about this project. And before we talk any longer about the science and the artworks without seeing them, I would say if I would open the floor to questions if there are. Otherwise, we walk over and have a look. No questions? Then let's go to the exhibition.